Chapter 25, the urinary system. In this first of 3 parts, I'll discuss how the kidneys can detox the body in a way that no magical milkshake or charm can do. Later in the term, we'll discuss how the liver can assist the kidneys in removing toxins from the human body, and we'll talk about what those toxins actually are. We'll also discuss the diabetic complication of kidney failure and why this is a common complication. Otherwise, in this first section, we'll mostly be discussing the basic anatomy of the kidneys. So I'll start with a gross anatomy, but then we'll move a bit more cellular and discuss the function of the functional unit of the kidneys, which is called the nephron. And we'll be paying closest attention to the first portion of the nephron, known as the glomerulus and the Bowman's capsule. These will be involved in filtering the blood, and we'll discuss how that filtration is regulated. The kidney has a few major functions. Its first major function is excretion. It can remove toxins and other unwanted chemicals from the human body. This will be a rather complicated topic. How does the kidney recognize what is toxic or just not needed versus something that I actually want, a nutrient, for instance? The kidneys are also involved in a number of homeostatic regulatory mechanisms, including the regulation of blood plasma volume. This will also be a huge topic in the next chapter. It's involved in the regulation of solutes, such as electrolytes and nutrients, and it's involved in the regulation of our body's pH. The kidneys are just one part of the urinary tract, the part that I'll be spending the most time discussing. But the urinary tract also includes the ureters, which connect to the bladder, which stores urine, and the urethra, which can excrete the urine. The process of excretion is called urination or micturition. We have to have a fancy name for everything. One thing that I find fascinating about the kidneys is that humans have basically had the same kidneys for tens of thousands of years, and yet these ancient kidneys are capable of excreting chemicals from the human body that have only been invented recently, such as AMPA, which is a breakdown product of glyphosate, which is the active ingredient of Roundup. Why would we have needed kidneys tens of thousands of years ago that were capable of removing Roundup from the human body fairly efficiently? And the answer to that is that the kidneys have no idea what is a toxin or a poison. So there are chemicals that could be invented 20 years from now, 200 years from now, 1,000 years from now if we survive that long, and the kidneys will still be able to excrete them from the human body. And this cartoon here represents the basic mechanism by which the kidneys can do this. They excrete everything. They take all of the small molecules out of the blood and remove them from the body, and then pump just the ones that we need back in. There are tens of thousands of different toxic chemicals and poisonous chemicals. Certainly, the kidneys can't know which ones are the bad ones. They can't have a list. It's absolutely not as simple as save the natural chemicals and get rid of the synthetic or unnatural ones. Let's see. Formaldehyde is a totally natural chemical, but not very good for the human body. Whereas aspirin is synthetic, and I'm going to need a couple of those after I'm done recording this lecture. So the kidneys get rid of everything, and then save just the nutrients that they know are beneficial for the human body, like amino acids, and certain electrolytes, and nucleic acids, and certain carbohydrates. It's a much smaller list of chemicals that are good for the human body. That's easier to remember than a list of things that are bad for the human body. So we simply get rid of everything. And then from that, we retrieve the things that we know are good, or that we know that we need, and everything else gets excreted. That's going to include toxins and poisonous chemicals, as well as excessive amounts of nutrients that we don't need. If I have excessive amounts of calcium or sodium, for instance, this can be excreted by the kidneys. So this, I think, is a very elegant mechanism for protecting the human body from very small dangers. The kidneys are retroperitoneal, meaning they exist behind the peritoneal cavity. Each kidney is surrounded by a fibrous capsule, an outer layer of connective tissue that marks the outer boundary of each kidney. They are then embedded within an adipose capsule, a bunch of fat that helps to pad the kidneys and protect them from damage. And this, in turn, is anchored into the body wall by fascia, or a type of connective tissue that helps to hold our organs in place. Some basic anatomy of each kidney. The hilum is the region where all of the tubes connect. This is a name we're going to see for more organs throughout the term. It's just a generic name for the area where tubes connect. I like to think of it as kind of the belly button region for any organ. The kidneys contain a number of sinuses, which are similar to the sinuses in the skull. In the skull, the sinuses were spaces within the bones that contained no bone tissue. The renal sinuses are spaces within the kidneys that don't contain any kidney tissue. They're not completely empty. They would be full of loose connective tissues like adipose or areolar connective tissue. The outer region of the kidney, just like any organ, would be called the cortex, whereas the deepest portion is called the medulla. In the medulla are a number of renal pyramids, these triangular shaped regions. Between the pyramids are columns where blood vessels and some other things travel. At the end of every pyramid is a little bump called a renal papilla. It's from here that urine will be dumped into a number of minor calyxes. From here, the minor calyxes can join up to form a couple of major calyxes, and the major calyxes join up to form a single renal pelvis. And as this tube exits the kidneys, we now start calling it the ureter. But it's not as if there's a dotted line or anything to tell us where the pelvis ends and the ureter begins. Within the pyramids, and out in the cortex as well, are a number of structures called nephrons. These are microscopic, but multicellular, and they're a series of tubes where the business of the kidneys occurs. So we could say that these nephrons are totally tubular, dude, and we're going to be spending a bunch of time talking about these tubes. Before we do, there's some more gross anatomy to cover, and that is that the kidneys receive about a quarter of our blood supply. That's about a liter of blood every minute that goes through the kidneys for cleaning. That blood enters the kidneys from the renal arteries. I'll highlight those here in addition to the renal veins. I'm going to keep both the arteries and the veins highlighted because they get the same names, but the blood is, of course, traveling in opposite directions. From the renal arteries, these split into the segmental arteries, which then branch into the interlobar arteries, which then split into the arcuate arteries, which arc around the sides of the kidneys. These arcuate arteries then turn into the cortical radiate arteries, and from here, teeny tiny afferent arterioles would lead to a whole bunch of different nephrons. The nephrons would filter that blood and then return a bunch of those components back to the bloodstream. When those components return, it becomes the cortical radiate veins, which become arcuate, interlobar, segmental, and finally renal veins, traveling in the opposite direction as the arteries. But luckily, they gave them the same names. Many anatomy textbooks skip over the segmental veins, but that's a mistake because a number of people do have segmental veins, but not everybody. In addition to tubes, nerves also connect to the kidneys. The sympathetic nervous system can adjust blood flow to the kidneys, either allowing more blood to be filtered or restricting blood flow and diverting it to our muscles when we're under stress. The sympathetic nervous system will also stimulate the release of the hormone renin. We're going to contrast that with the digestive enzyme renin when we get to the digestive system. So make sure your spelling is correct here, because it'd be easy to spell a completely different molecule. But that's the gross anatomy of the kidneys. Next, we're going to move on to the physiology. Next, I'm going to discuss the functional unit of the kidneys, the nephron. Each nephron begins with a tiny ball of capillaries known as a glomerulus. I know it doesn't look like it's pronounced that way from its spelling, but it is. And each glomerulus is enclosed by a Bowman's capsule. That's the beginning of our nephron. Here's my cartoon of what these two things look like: a squiggly ball of capillaries and a cap around them. If we zoom in, we'll notice that these capillaries are special. They are a fenestrated endothelium, one of the types of capillaries that last quarter I said I wouldn't discuss until this quarter. So here we are. These capillaries are different from regular capillaries in that they have tiny little pores, which makes them more permeable than your average capillary. And this helps them to do their job, which is filtration. All of the material that filters out of these capillaries is captured by the Bowman's capsule, which is the beginning of our nephron. The liquid component of blood is called plasma, but as this plasma leaks out of the fenestrated capillaries into the Bowman's capsule, we now call that fluid filtrate. It's from this filtrate that the nephron will reabsorb necessary nutrients, leaving everything else to be excreted, which will be excess nutrients, toxins, poisons, and other waste molecules that the body has produced. Blood pressure in the glomerulus is high because the afferent arteriole, the artery leading to the glomerulus, is a bit larger than the efferent arteriole, the artery leading away. And this will mean that a bunch of fluid will be forced out of the capillary bed into the Bowman's capsule. And this fluid will be then collected by the nephron. From here, the fluid travels to the proximal convoluted tubule, a single squiggly tube. This connects to the loop of Henle, which dives down deep into the medullary region and then back up towards the cortex, where it reaches the distal convoluted tubule, another singular squiggly tube. From the distal convoluted tubule, fluid would next travel to the collecting duct, which is the last part of our nephron. Here in this cartoon, it's supposed to look like multiple distal convoluted tubules can each connect to a single collecting duct. To review really quickly, the cortex is the most superficial region of the kidney, while the deeper portion is called the medulla. The majority of nephrons are called cortical nephrons because they exist primarily within the cortex of the kidneys. Some nephrons are called juxtamedullary nephrons. These start close to the border between the cortex and the medulla, and the loop of Henle of these nephrons dives deeper into the kidneys. And it's the depth at which the loop of Henle travels that will be very significant when it comes to how much is reabsorbed. We have a few more bits of microscopic anatomy to discuss, however, and those two things are blood vessels. So blood entering the glomerulus came in through the afferent arteriole to the glomerulus, and then whatever didn't get filtered left via the efferent arteriole, which then connects to another capillary bed called peritubular capillaries. These are named because they wrap around the tubules. These peritubular capillaries would ultimately connect to a cortical radiate vein, which would return blood back to the general circulatory system. So that's the basic anatomy of the kidneys as well as the nephrons. Next, I'm going to discuss how the kidneys are involved in regulating blood pressure. Last quarter, I told you that when the kidneys become hypoxic, they release the hormone erythropoietin, which can trigger red blood cell production in the bone marrow. To regulate blood pressure, however, we need to do something a little more quick. We need to regulate fluid volume. That's going to involve the processes of filtration, reabsorption, and secretion. For starters, all of the fluid is going to be in the bloodstream, but at the glomerulus, some of this fluid will enter into the nephrons to become filtrate. So we have now reduced some of the fluid in our blood supply. This will, in effect, reduce blood pressure. However, there's a bunch of goodies in this filtrate that I don't want to lose. So we're going to reabsorb a bunch of those goodies. And those goodies are solutes. So as we reabsorb solutes back into the bloodstream, water is going to follow, which increases the volume of our blood, raising blood pressure. Furthermore, if I secrete anything, actively move molecules from the blood to the filtrate, then water will tend to follow those as well. Let's start with filtration. The amount of fluid that is filtered at the glomerulus is regulated by two cell types: our fenestrated endothelium, or the outer borders of the capillaries, and another cell type called podocytes, or foot cells, which sit on top of these capillaries. Anything that wants to get out of the bloodstream and into the nephron has to get past these two cells. Podocytes are a highly specialized type of epithelial cell. These surround the capillaries. The podocytes are named because of their dendritic extensions called foot processes. These are long and skinny and get up right next to the foot processes of the next cell. In this picture, we've got some of the podocytes highlighted in blue and another big one highlighted in green. So hopefully you can see where the little blue foot processes come up right next to the green ones. They don't quite touch, however. They leave a little bit of a gap between the foot processes, known as filtration slits. The only molecules that are going to be able to leave the bloodstream and be filtered by the kidneys have to be small enough to fit between these filtration slits, as well as fit through the pores of the fenestrated endothelia. They must also go through a shared basement membrane, and these three structures make up what is known as the filtration membrane. This should look fairly similar to the respiratory membrane of the last chapter, but now we're in a different organ serving a different purpose. Once molecules pass these three layers, they would then wind up in the Bowman's capsule, and we would call them filtrate. Only molecules, again, that are small enough to fit through the pores and the slits would be able to leave the bloodstream and wind up as filtrate. Really large molecules, like albumin and other large proteins found in blood plasma, would not wind up inside the nephrons, nor would really large things like red blood cells or white blood cells. Only small molecules, like water, and glucose, and electrolytes, and other nutrients and toxins, are capable of being filtered by the kidneys. So it's only small molecules that can pass out of the bloodstream to become filtrate. That includes nutrients like glucose and amino acids, waste products like urea, and toxins and poisons like endotoxin A or lipopolysaccharide. These things could leave the bloodstream and be filtered. But the really large things like proteins, such as the albumin that I just mentioned, and antibodies as well. These are going to stay in the blood because they're not only too large, but amino acids contain R groups, some of which are going to be negatively charged, some of which are positively charged, and the negatively charged R groups would be repelled by the negatively charged basement membrane. So that's what helps to keep large things in the blood, but allow small things through. Next, I'm going to be talking about reabsorption, which has to be highly efficient because the kidneys filter the blood plasma 60 times per day. That means if I had 1 molecule of glucose that got filtered and it was reabsorbed, it would just get filtered again and reabsorbed and so on 60 times per day if it wasn't used by any other cells in the human body. If I had to use 1 molecule of ATP to reabsorb that 1 molecule of glucose 60 times, I don't generate 60 molecules of ATP by burning that glucose. I would be losing energy. So the kidneys must be highly efficient in their task. This process begins at the glomerulus, where it's a strictly mechanical process. Small things get through the pores and wind up as filtrate. The rest of the nephron is going to be a little bit more complicated. We're going to have to discuss some molecular biology when it comes to reabsorption. But that's for the second portion of the chapter. For right now, we're going to stick with filtration. These capillaries are highly efficient at filtering blood, not only because of their tiny little pores, but because of the high blood pressure, due to the fact that the afferent arteriole, let's see if we can find one here, is a little bit larger than the efferent arteriole. This is going to mean that the pressure inside the glomerulus is higher than in your typical capillary bed. Before I move on, it's very important to note that the pores in the fenestrated endothelia, as well as the filtration slits between the podocytes, be of uniform size. If they were too small, waste products and toxins would not get filtered and instead would build up in the bloodstream. But if they were too large, then too many nutrients and other things would get lost into my urine. One of the dangers of diabetes is it can lead to damage of tiny blood vessels, especially the tiny capillaries of the glomerulus. This can lead to scarring, which blocks the pores in the slits, which reduces kidney function. And this means that toxins and waste products will build up in the body. What I find fascinating is that ketogenic diets have been shown to reverse this damage. And I find this fascinating and amazing because it's very cheap to put a patient on a ketogenic diet. We can measure this by doing what's called a creatine clearance test, or just by looking at how much albumin is in the urine. With healthy kidneys, there shouldn't be any, because this blood plasma protein is too large to fit through a healthy filtration slit. Healthy glomeruluses should be producing about 180 liters of filtrate every day. Now that sounds like a lot, but there's significantly more blood traveling to the kidneys every day. Only about 10% of the fluid that enters the glomerulus gets filtered. Out of that, though, we need to reabsorb nearly all of it. 99% of our filtrate gets reabsorbed and returns to the bloodstream. Only 1% of that 10% actually becomes urine in a healthy pair of kidneys. It's important that my glomerular filtration rate be just right. If the GFR was too high, let's say those filtration slits and pores in the endothelia had become damaged and were too wide, I would be losing too many nutrients and other molecules to my filtrate, and the nephrons can only reabsorb so much. If I overloaded them, I would be losing too much from my bloodstream. Conversely, if those pores and slits became clogged, my GFR would be too low. I wouldn't be filtering enough, and this could lead to toxins building up in the bloodstream. So, we're going to need to keep GFR within a narrow window, and that's going to mean homeostasis. And that means we have a number of homeostatic mechanisms involved here. Not just one, but several. And by several, I mean 3. We've got renal autoregulatory mechanisms, we have neural controls, and we've also got hormones. So let's talk about these 3 one at a time. The first are renal autoregulatory mechanisms. Autoregulatory means that the kidneys can regulate how much filtration is occurring themselves. This is in contrast to the other 2 mechanisms, which will require signals from outside of the kidneys. So inside of the kidneys, we can measure blood pressure in the afferent arteriole, and this can lead to changes in the amount of filtration that occurs. We can also measure flow-dependent feedback, which is, in effect, measuring the pressure inside the Bowman's capsule. This is a measurement of how much filtration is occurring. And in a classic negative feedback loop, too much filtration will lead to an elevation in pressure inside the Bowman's capsule, which can lead to a reduction in the amount of filtration. Filtration can be regulated by the size of the afferent arteriole. Constricting the afferent arteriole will reduce blood flow to the glomerulus, while dilating it will increase pressure in the glomerulus. Next up is neural regulation. The sympathetic nervous system can shut down the kidneys. With stress or in anticipation of exercise, neurons will trigger the constriction of the afferent arteriole. So little or no blood will reach the glomerulus. And if nothing reaches the glomerulus, nothing can be filtered. Before I move to the third mechanism, I want to tell you a little story. During some of my martial arts tests, we started off having tests that were a couple hours long, and as I worked my way up through the ranks, these tests became 6 hours, 8 hours, 8 hours for 2 days, 8 hours for 3 days. The tests were really, really long, and we weren't allowed water breaks. So the first time that I took an 8-hour test, I asked one of my classmates, who was a medical doctor, what are we going to do? How are we going to perform in this physical activity for 8 hours without drinking water? And he said, drink all the water that you're going to need right now. And I was afraid, won't that make me have to urinate? 20 minutes from now? We're not allowed bathroom breaks either. What am I going to do when I have to pee? And he said, oh, you're not going to have to pee. Don't you worry. So I drank a couple of gallons of water right up front, and sure enough, I never had to pee. I was stressed right away, and my sympathetic nervous system just shut down my kidneys. All of my blood was going elsewhere. It was needed in my arms and legs and some other places to deal with all of the exercise that I was doing. So that was my first-hand account to let you know that the sympathetic nervous system is very efficient at shutting down blood flow to the kidneys. And if you're exercising, your body really shouldn't be producing that much urine, if any at all. Lastly is hormonal regulation. Blood flow to the glomerulus, and hence GFR, is also regulated by the hormones renin and angiotensin. And these hormones are produced in response to a number of humoral stimuluses. Humoral, of course, means in our body fluids. So the kidneys can detect blood pressure, blood osmolarity, and specific chemicals in the blood like carbon dioxide. These chemicals can then trigger the release of hormones from a structure called the juxtaglomerular apparatus. The juxtaglomerular apparatus, or the JGA, is composed of 3 major components, 2 of which will be on your test. The first is the macula densa. This region here of the distal convoluted tubule. Now, this distal convoluted tubule comes really close to the Bowman's capsule, and yet it's still called the distal convoluted tubule. Remember, it's the proximal convoluted tubule that's connected to the glomerulus, but the distal tubule can come close for the same reason that the most distal part of your arm, your fingertips, can get really close to your shoulders. Just because your fingertips are touching your shoulders doesn't make them proximal. They are still the most distal region of your arm. Similarly, the distal convoluted tubule winds backwards to travel close to the glomerulus. And here there is a region of thicker cells known as the macula densa. And it's here that chemo and osmo receptors are located, detecting the osmolarity of our filtrate, as well as detecting specific chemicals found within that filtrate. The JG cells are located on the afferent arteriole, and these are mechanoreceptors. These are the ones that detect blood pressure, the same ones that I talked about in our first mechanism, the autoregulatory mechanism. In addition to measuring blood pressure and triggering vasoconstriction or vasodilation of the afferent arteriole, changes in blood pressure can also trigger the release of renin from the JGA. The third part of our JGA, and the part that's not very well understood, and hence I will mention it but not test you on it, are mesangial cells found next to the glomerulus. The JGA functions to release the hormone renin, and these three parts of the JGA will trigger the release of renin for three different reasons. The first is reduced stretch of the JG cells, which is another way of saying a drop in blood pressure. If our blood pressure drops, we don't want to be producing too much more filtrate. So this drop in blood pressure, measured by these JG cells, will trigger a release of the hormone renin. Renin can also be released when the macula densa becomes activated, and it does so when it detects a drop in sodium levels, for instance. And third, the sympathetic nervous system can also trigger the release of renin. So, number 1 and number 3 not only triggered vasoconstriction of the afferent arteriole, they also trigger the release of renin from the JGA. And both constriction of the afferent arteriole and the release of renin are going to lead to a drop in GFR. And a drop in GFR means that fluid is not going to be lost, but instead stay in the body, and this keeps blood pressure high, which is especially important when we're exercising. Hence, the sympathetic nervous system shuts down the function of the kidneys. The effects of renin are indirect. It will activate the hormone angiotensin 1, which in turn activates the hormone angiotensin 2. Perhaps you remember this from our discussion of the respiratory tract. If not, you might go back a ways and re-look at those slides. It's angiotensin 2 that has effects on GFR and blood pressure. Angiotensin 2 causes body-wide vasoconstriction, and as you remember from BI 232, vasoconstriction uniformly increases blood pressure. This also leads to vasoconstriction of the afferent arteriole. And when we constrict the afferent arteriole, that's going to lower blood flow to the glomerulus, lowering GFR. And by lowering GFR, I am instead saving most of the fluid for my blood rather than let it be filtered to become urine. Angiotensin 2 also stimulates the adrenals to release the hormone aldosterone, which we'll talk about later. And it also has other effects in the central nervous system that are similar, triggering the release of the hormone ADH, stimulating thirst, and increasing sympathetic tone. So those are the three mechanisms by which the rate of filtration is controlled. I talked about autoregulatory mechanisms, the neural mechanism, and the renin-angiotensin pathway. I didn't quite get through the renin-angiotensin pathway completely. We'll have to come back and discuss hormones a bit more after we get a little bit more physiology. But for now, this would be a really good spot to take a break, grab a drink, get some rest, and come back to the second section when you're ready. And welcome back. In this second part of 3 parts, we'll be learning about the molecular biology behind the function of diuretics, one of the most commonly prescribed medications. We'll focus mostly on the physiology of the human body, but in doing so, we'll learn how these drugs work and how they can be used to treat hypertension, and we could also discuss how these types of drugs could be abused in sports. We'll talk about the basis as to why a high sodium diet may raise blood pressure. It's not going to be effective at doing so in all of your patients, but it will in some of your patients, depending on their race. And this will involve a number of homeostatic mechanisms. We'll also talk about the loop of Henle today, and this very confusing bit of physiology called counter-current exchange. But it's a very elegant bit of physiology, which is important because we're filtering the blood 60 times per day. That means I'm losing a water molecule and reabsorbing it 60 times per day. I need to be very efficient at how I reabsorb that water molecule. Otherwise, that would take a lot of energy to clean my blood. To do all of that, I'm going to be discussing the processes of absorption and secretion. And next, I will be assigning functions to the other parts of the nephron that we haven't covered yet. The proximal convoluted tubule, loop of Henle, the distal convoluted tubule, and the collecting duct. And I'll be introducing a couple of hormones that function at specific places here. In this part of the lecture, we're going to spend most of our time zoomed way in, down to the cellular level, looking at how nutrients get reabsorbed from the filtrate over here inside of the nephron into the bloodstream, which would be outside of the nephron, into those paratubular capillaries. To do that, molecules must cross one of the epithelial cells that makes up the nephron. These are usually a simple cuboidal epithelium. Organic nutrients are always fully reabsorbed. By organic, I of course mean molecules that contain carbon atoms, like glucose and amino acids. And by fully reabsorbed, I mean I reabsorb as many of those as possible, never anything less than that. But I could always eat excessive amounts of amino acids or glucose, and the excess amounts would not be reabsorbed. I'll have an illustration for that coming up. On the other hand, the reabsorption of water and electrolytes is tightly controlled by a number of hormones. So you're going to have more physiology to learn about when it comes to the reabsorption of these items. The reabsorption of all nutrients is driven by a sodium gradient, and this sodium gradient is set up by the sodium-potassium pump, the same protein that we learned about last quarter when we learned about neurons. This protein can be found on the basolateral side of these epithelial cells in the nephron. And by using ATP, this protein will pump sodium ions out of the cells into the extracellular fluid. This means that sodium from the lumen of the nephron would want to enter these cells down its concentration gradient. From the extracellular fluid, sodium ions would be absorbed into the paratubular capillaries. And if nothing was moving, we would eventually build up high levels of sodium over here in the extracellular fluid side. But that doesn't happen, of course, because blood is flowing. It's going to be carrying those sodium ions away. So our sodium-potassium pump can keep pumping sodium out of these cells. This gradient is used to drive the reabsorption of water and other nutrients. Water will follow passively by osmosis with the assistance of some proteins called aquaporins on the apical surface of these epithelial cells. I'll come back to the aquaporins in a bit. Instead, for right now, we're going to focus on the reabsorption of nutrients. To bring any other nutrient, like glucose, into the bloodstream, we're first going to carry it across the apical surface into the cells. We're going to be able to do this by using the sodium gradient. Sodium wants to rush into these epithelial cells because the sodium has been pumped out of them on the basolateral surface. Sodium ions can't just diffuse across the plasma membrane, however. They carry a charge, and the plasma membrane has that lipophilic core. So to get sodium down its concentration gradient, it'll have to go through some sort of ion channel or other transport protein. So we're going to have co-transport proteins that can move a nutrient like glucose in addition to a sodium ion from the lumen of the nephron into these epithelial cells. This is called facilitated diffusion. These co-transport proteins don't use ATP, they're using the energy of the sodium gradient to transport both sodium and glucose into the cell. And as long as the sodium gradient is bigger than the glucose gradient, we'll be able to move glucose against its concentration gradient. The same way that wind, in order to get past a windmill, has to push on the blades and turn them, we can get it to do a little bit of work for us. So glucose is going to wind up inside of these cells. We can then allow it to diffuse out of these cells, where once again bulk flow helps to pull it away. Therefore, we don't have an ever-increasing gradient for glucose. We only have so many sodium-glucose co-transporters, however. So if we have too many glucose molecules, I won't be able to reabsorb all of them. And that's because the glomerulus is continually filtering blood, making new filtrate, which will push the old filtrate towards the distal end of the nephron. So as we reabsorb glucose, some of the other glucose molecules might get pushed down the nephron and not get reabsorbed. We're going to call this the transport maximum, and it's based off of the number of glucose transporters that exist on the apical surface of these cells here in the nephron. If I have more glucose in the filtrate, it could exceed the transport maximum, and some of that glucose would be lost. Instead of being reabsorbed, it would wind up in urine. Other molecules, especially ones like urea, creatinine, and uric acid, do not contain transporters. So anything that's not a nutrient, like a waste product or a toxin that is small enough to be filtered, when it winds up in the nephron, it's just going to stay there and never get reabsorbed. And it'll eventually be pushed all the way to the end to become urine and be excreted from the body. This is how the nephrons clean the blood. They remove everything that's small enough at the glomerulus, and then only reabsorb the things that we know we like, such as glucose, amino acids, and nucleic acids, and certain electrolytes. So that's a basic overview of how reabsorption works. We have the sodium-potassium pump on the basolateral side of the cells, and a sodium nutrient co-transport protein on the apical surface that's engaged in facilitated diffusion. Only the sodium-potassium pump was engaged in active transport. That's where we used the ATP to set up the sodium gradient that we use to drive the reabsorption of all of the other nutrients. Next, we're going to go into more detail about what gets reabsorbed where. First, everything that winds up in filtrate in the Bowman's capsule will travel to the proximal convoluted tubule, and it's here that the most reabsorption occurs. Nearly 70% of everything that's going to get reabsorbed will be reabsorbed in the PCT. That includes all of our organic nutrients, like glucose and amino acids, as well as a bunch of water and electrolytes, including sodium. All of these items that are reabsorbed wind up in the peritubular capillaries to be returned to our general circulation. Whatever doesn't get reabsorbed in the proximal convoluted tubule will next wind up in the loop of Henle. The loop of Henle has a descending limb where filtrate is traveling down deeper into the kidney, and an ascending limb where the fluid takes an about face and travels more superficially. In the descending limb, water is primarily reabsorbed, and in the ascending limb, we primarily reabsorb solutes, such as sodium and some other electrolytes. This is a little bit of an oversimplification, but definitely good enough for our needs. Some other electrolytes get reabsorbed in the distal convoluted tubule, and generally speaking, the reabsorption of these electrolytes is regulated by different hormones. For instance, you all remember parathyroid hormone from BI 231. This can turn on the reabsorption of calcium ions from the filtrate back into the bloodstream. If we had excessive amounts of calcium in the blood, this hormone would no longer be produced. Instead, calcitonin would be shutting off calcium reabsorption in the nephrons. Hence, we would be losing more calcium to our urine in that case. We'll learn more about aldosterone coming up, but this can help to regulate the reabsorption of sodium and hydrogen ions, and some other electrolytes are also reabsorbed here. In the collecting duct, we primarily reabsorb water, and this will be under the control of another hormone called antidiuretic hormone. It's possible to secrete molecules as well. This would mean we would take molecules from the blood and transport them into the filtrate. We do this to get rid of things that the body knows that we need to get rid of. For instance, we can get rid of excess hydrogen ions in order to help control the pH of our blood. However, we mostly rely on simply not reabsorbing things to get them out of the human body. For instance, 3 major waste products get filtered at the glomerulus and then are not reabsorbed. Hence, we can excrete large quantities of urea, creatinine, and uric acid. These are waste products. They're not toxic, they're just not useful to the human body, so we don't want them building up in high concentrations. And the kidneys are able to effectively remove these from the bloodstream as they are produced. Next, we're moving on to the loop of Henle, and here we need to talk about 2 bits of physiology: counter-current exchange and the counter-current multiplier. Counter-current exchange will occur as filtrate and blood flow in opposite directions from one another, and this will greatly increase the efficiency of reabsorption. We'll illustrate that in a moment. First, though, we need to talk about the counter-current multiplier. This uses ATP to set up a concentration gradient within the kidney for solutes. Out in the cortex, solute levels are low, roughly as concentrated as anywhere else in the body. But as we dive deeper into the kidneys, the amount of salt goes upwards. Let me go back to my nephron now, and I'll superimpose the solute gradient. Out here in the cortex, solute levels are low, but deep down in the kidneys, the solute levels are high. And the only parts of the nephron that get down deep into the medullary region is this loop of Henle that we'll talk about now, and the collecting duct, which we'll get to in a moment. The filtrate that has been produced at the glomerulus will travel to the proximal convoluted tubule, where most everything gets reabsorbed. And then next, it would travel down to the loop of Henle and the descending limb. And this part of the loop of Henle is permeable to water. It contains aquaporins, which are proteins in the plasma membranes of these cells that allow water to move passively down its concentration gradient. Because the filtrate that we've made is fairly low in solutes, and it's now traveling to a region where we have high levels of solutes outside of the nephron, water would want to move down its concentration gradient, or out of the filtrate. And the aquaporins allow it to do so. So in the descending limb of the loop of Henle, we can reabsorb water passively. This takes no energy. We're simply relying on the concentration gradient of the solutes that we set up earlier with the countercurrent multiplier. As water leaves the filtrate, however, and gets reabsorbed, it leaves behind the solutes. So the filtrate becomes more concentrated. It should be roughly as concentrated as the solute concentration on the outside. But now this fluid gets pushed up the ascending limb of the loop of Henle, and it reaches a region more superficial, where the solute concentration outside is now lower. And the ascending limb of the loop of Henle is permeable to solutes like sodium. So now we have a situation where solute levels are high inside the loop of Henle, but low outside. And because we now have sodium channels and other ion channels, these solutes can leave the nephron to be reabsorbed. Once again, this is passive and requires no energy. So we've reabsorbed water in the descending limb and solutes in the ascending limb. So what is countercurrent about this? Well, the blood is flowing in the opposite direction. The capillaries that travel right next to the loop of Henle are called the vasa recta, and blood is flowing in the opposing direction. So it can pick up the solutes over here, and as those solutes, which are now in the bloodstream, travel to the opposite side, they help to attract water, aiding in the reabsorption of water from the descending limb. And that's the countercurrent portion. The filtrate and the blood are flowing in opposite directions. The countercurrent exchange mechanism allows us to reabsorb about half of the water from the filtrate, as well as 2/3 of the sodium and chloride and some other electrolytes. If we simply relied on diffusion, allowing the filtrate and the extracellular fluid to reach equilibrium, I could either get half of the water or half of the sodium chloride, not half of the water and more than half of the sodium chloride. The countercurrent exchanger was passive, but it did rely on the countercurrent multiplier, which does take energy. I had to use some ATP to pump that sodium to the medullary region in the first place. But once it's there, I can reuse it over and over again to keep driving the reabsorption of water, which concentrates my filtrate, which then allows me to reabsorb electrolytes out in the cortex. Some of my students like to skip the lectures and instead go to Google to try and answer their homework questions. And they're going to be learning all about how refrigerators work when they type in countercurrent exchange. You, on the other hand, need to tell me about how countercurrent exchange works in the loop of Henle within our kidneys. To help you with that, I've got a goofy little illustration here, where I'm trying to represent 6 individual segments of a nephron, here in yellow, and 6 corresponding segments of a capillary, the vasa recta, right next to the loop of Henle. And filtrate is flowing in the nephron to the left-hand side of the page, whereas blood is flowing to the right-hand side of the page. And we're going to start off with 4 ions of sodium in these 3 segments of the nephron down here. And we're just going to allow diffusion to occur. We'll wind up with 2 sodiums in each of these regions of the capillaries and the blood vessels. This requires no energy. But stuff is moving, so we're going to push these sodium ions to the left in the nephron and to the right in the capillary. And what this is supposed to represent over here is that I just reabsorbed 2 sodium ions back into my general circulation. And down here in the nephron, I filtered 4 new ions of sodium. You'll notice we still have a concentration gradient at every region between the nephron and the blood vessel. So sodium is going to be diffusing down its concentration gradient here, here, here. Whoops, not there, not right now, but it will be doing so over here. And we'll reach a new equilibrium. Push everything to the left and to the right. I've reabsorbed some more sodium. I have filtered out some new sodium ions here. And now I still have a concentration gradient at every one of these regions. Here, 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 and here. So sodium is still going to be diffusing down its concentration gradient. And now we'll push things to the left once again. I still have my sodium concentration gradient at every region. So as long as fluid is flowing in opposing directions, I will always be reabsorbing sodium from the filtrate into the bloodstream. That is the power of countercurrent exchange. We can greatly concentrate all of the sodium in one region, my blood, and I'm not losing very much of it to my urine over here, thanks to the opposing flow of filtrate and blood. And if I kept on doing this forever, I would keep on having a concentration gradient for every region between this nephron and this vasa recta capillary. So I've reabsorbed a whole bunch of water and a whole bunch of electrolytes in the loop of Henle, in addition to reabsorbing a whole bunch of nutrients, water, and electrolytes in the proximal convoluted tubule. Therefore, very little is going to reach the distal convoluted tubule. Only about 1/5 of our original volume will wind up in the DCT. But what does reach the DCT is important. We're going to reabsorb a number of things here, but we're going to do so very carefully. It'll be tightly controlled by a number of hormones. We make some final adjustments to the filtrate here at the distal convoluted tubule. One of the most important ones is the reabsorption of sodium here under the control of the hormone aldosterone. Aldosterone is produced in the adrenal glands just above the kidneys, and it will activate the sodium-potassium pump here in the distal convoluted tubule. Therefore, more sodium will be reabsorbed from the filtrate back into the bloodstream. We can talk about what reabsorbing more sodium does in a moment, but for now let's focus on what's happening to potassium, because this is going to be more important for your patients. With more aldosterone will lead to more activity of the sodium-potassium pumps. We'll be reabsorbing more sodium, but for every 3 sodium ions the sodium-potassium pump moves, 2 potassium ions are going in the opposite direction, out of the body into the filtrate to be lost in urine. Therefore, with excessive aldosterone can be excessive activity of the sodium-potassium pump, and we could lose too much potassium from the body, which is a condition called hypokalemia. And potassium is a very important ion. We talked about it a lot last quarter when we talked about neurons. But many cells have sodium-potassium pumps that set up electrical gradients across their plasma membrane. Even those osteocytes that we talked about a long time ago had electrical gradients across their plasma membrane that they use to detect stress within bone tissue. So we don't want to lose too much potassium because it could mess with our ion gradients across cells everywhere in the body. But we do especially worry about neuronal function up in the brain when our electrolyte levels are imbalanced. Let's go back to our major nitrogenous wastes of urea, uric acid, and creatinine. These molecules are small enough to be filtered at the glomerulus over here, but because they contain no transport proteins anywhere along the nephron, they won't get reabsorbed. But as water and other nutrients are reabsorbed as this filtrate travels through the neuron, we're leaving behind these waste products. Hence, the waste products become concentrated. And at the end of the collecting duct here, we're going to have very high levels of urea, uric acid, and creatinine, as well as any other molecules in the body that our nephrons don't recognize as nutrients, such as toxins. The last section that we have to cover is the collecting duct. And now I need to focus a little bit more on these proteins that I briefly mentioned earlier, the aquaporins. These are transmembrane proteins that allow water to move across the plasma membrane. Water can diffuse across the plasma membrane on its own, but it's difficult because water molecules, being polar, don't really like to travel through the non-polar lipid tails here of our phospholipid bilayer. So to speed up the movement of water down its concentration gradient, we can use these aquaporin proteins, which simply allow water to move down its gradient. There is no pump for water anywhere in the human body, so there's no ATPase that can move water against its concentration gradient. Therefore, we're always going to have water moving down its concentration gradient, and we must rely on osmosis. But aquaporins can help osmosis to act as quickly as possible. If I didn't have any aquaporins, the filtrate that left the ascending limb of the loop of Henle would have been fairly dilute, because I've reabsorbed a bunch of electrolytes. And if this reached the collecting duct and nothing else happened, then it would become dilute urine. Aquaporins, however, can change this. If I move aquaporin proteins to the apical surface of the cells in the collecting duct, we're going to reabsorb a lot of water. And this is going to be under the control of the hormone ADH, or anti-diuretic hormone. This hormone is produced by the hypothalamus, and it signals the movement of aquaporins into the collecting duct. And that will make the collecting duct permeable to water. And as we remember, the collecting duct is traveling deeper into the kidney. Hence, it's moving to regions of high solute concentration. So as long as there are aquaporins here, water will leave the nephron and be reabsorbed. And as that happens, this will effectively concentrate whatever is left over. Those would be the solutes. And this will make concentrated urine. As long as we are producing ADH from the brain, we should be reabsorbing 99% of the water that gets filtered up here at the glomerulus. The brain normally produces ADH all the time because we like having water. The only time that we wouldn't be producing ADH is if we drank excessive levels of water, which would drop our solute concentration around the hypothalamus. Or, as you may remember, drinking alcohol can do the same thing, because alcohol will block the production of ADH. Hence, we will not have aquaporins functioning in the collecting duct. And this means that any fluid that enters the collecting duct will become urine, and we will be losing excessive amounts of water to our urine if we've been drinking alcohol. Before I move on, I want to point out that ADH has other names like vasopressin, arginine vasopressin, or argipressin. I will do my best to keep calling it antidiuretic hormone. It also has other jobs that have nothing to do with water retention, but we don't really have time to go into those right now. Before we move on, I need to point out that there's a form of diabetes called diabetes insipidus that few people know about. And it's rather odd because it has nothing to do with sugar in the bloodstream, which we usually think of when we hear the disease diabetes. And in fact, diabetes was originally named because patients would overproduce urine, which could lead to dehydration. Diabetes insipidus led to excess urine production because these people had a lack of the ADH hormone or faulty ADH receptors. Either way meant that aquaporins would not be functional in the collecting duct. Hence, these people cannot reabsorb water here, and they will continuously produce dilute urine, even when it means losing too much water from the body. Later, we'll contrast this with the more common form of diabetes, diabetes mellitus, which is the one that involves excess levels of glucose in the bloodstream. But we'll get to that in a moment. Let's stay focused on water reabsorption. In the collecting duct, we could either turn on or turn off the aquaporins. We also reabsorbed water in the proximal convoluted tubule and the descending limb of the loop of Henle. There were aquaporins here as well, but these aquaporins are always there. It's only in the collecting duct where we can turn things on or off. Hence, we call water reabsorption that happens in the proximal convoluted tubule and the loop of Henle obligatory. We have to reabsorb water at these locations, and we never change the amount. Water reabsorption in the collecting duct is referred to as facultative. I can reabsorb more water here or less, depending on how hydrated I am. That's important because we're filtering our blood 60 times per day. We would have to drink a lot more water, 60 times more water than we do now, if I wasn't reabsorbing almost all of it every time it got filtered. Hence, my body is usually producing ADH, unless I just drank a whole bunch of water. It's here that I'd like to cover the link between a high sodium diet and high blood pressure. And this has to do with the fact that I can't pump water. To get water to move, we must first pump electrolytes, like sodium, and then water can follow by osmosis, assuming that there are aquaporins present. So as we reabsorb more sodium from the filtrate back into the bloodstream, more water is going to follow, especially at the collecting duct. And as more water re-enters the bloodstream, that's going to mean a higher volume of blood, which means higher blood pressure. So, a high sodium diet can lead to high blood pressure, but it doesn't always. That's probably because if you have high levels of sodium in the blood, that would lead to higher levels of sodium in the filtrate. And if we're losing more sodium in our filtrate, it's going to pull water along with it. But some people are better capable of reabsorbing sodium from their filtrate back into their bloodstream than others. And for these people, cutting sodium intake can greatly lower their blood pressure. This is especially true for African American men, but not Africans. This likely has to do with the slave trade, where many slaves taken from Africa were forced onto ships in horrible conditions, and many of them died along the way because they weren't given adequate water. For those who were capable of reabsorbing even more sodium out of their filtrate, that meant that they were going to be reabsorbing more water as well. Hence, they would be losing less water to their urine, which helped to protect them from dehydration. So, Sad as this history is, that is probably why African Americans will benefit more from a low sodium diet than people of other races. Now, potassium also plays a role here, but I am not going to be spending time discussing potassium's effects on blood pressure. Some of your patients who go on low sodium diets may not see any effect. Nevertheless, most of us eat way too much sodium in our diet. The average American diet has about 3 grams of sodium per day, and we really only need 1.5. So most of your patients are going to have excessive levels of sodium in their blood. For many of us, though, that will also lead to excess levels of sodium in our filtrate, which winds up in urine, pulling some water along with it. So sodium won't always lead to hypertension. That's because of the function of our kidneys. But in some folks, this excessive sodium can contribute to hypertension because water follows sodium. Next, I need to discuss kidney stones. As we reabsorb more water from the filtrate, this leaves behind many electrolytes. And many of those electrolytes are minerals which can form crystals. They don't crystallize when dissolved in water, but as we remove water from the filtrate, we're now concentrating the solutes. And if they start forming crystals, these crystals can cause pain as they travel through the rest of the urinary tract. Our urinary tract is designed to allow fluids to flow freely, but kidney stones or renal calculus can block these tubes or cut them up as they travel through the tubes, leading to bleeding and pain and inflammation. Certain foods can increase the ability of kidney stones to form, such as high fructose corn syrup and grapefruit juice. Other electrolytes are more prone to crystallize than others, like sodium. Conversely, the more fluids that we drink, the more of that fluid will wind up here in our filtrate, which will tend to form more dilute urine, hence less likely to have crystals forming. So dehydration is one of our big risk factors for kidney stone formation. It's important for patients who are prone to getting kidney stones to make sure that they are properly hydrated at all times. But there's other things in our diet that we can reduce or avoid to decrease the chances of kidney stones forming, such as this list here. But this would be a good spot to take another break. We talked about the basic functions of the rest of the nephron. 70% of all of our nutrients and electrolytes were reabsorbed in the proximal convoluted tubule. The loop of Henle is where we had counter-current exchange occurring, where we reabsorbed water in the descending loop and solutes in the ascending loop. And those were reabsorbed into the vasa recta. In the distal convoluted tubule, I focused primarily on the function of aldosterone, turning on the sodium-potassium pump, and that sodium could be reabsorbed into the peritubular capillaries. Lastly, in the collecting duct, ADH activated aquaporins, which allowed water to be reabsorbed, thanks to that high solute concentration deep in the medullary region of our kidneys. But now would be a good time to take a break, grab a drink, have some rest, and come back later. Okay, welcome back. This is the third of 3 parts about the urinary system. And in this last bit, we will be putting it all together. We'll talk about the function of diuretics in more detail in this part. We'll also learn about how dialysis works, both hemodialysis and peritoneal dialysis. I'll move on to discuss stem cell therapy and how we can grow bladders in a lab. And lastly, we'll learn how to pee. So you've been waiting for that for hours, it seems like. So to do that, we've got a little bit more physiology to cover. So I will summarize all of the different factors that are involved in regulating how much urine is produced. That will mean reviewing GFR as well as reabsorption and secretion. And then we'll talk about how urine is produced and excreted from the body. We have covered two major mechanisms by which the body can regulate how much urine is produced. It can control the glomerular filtration rate, and it can control reabsorption. GFR is controlled by an autoregulatory mechanism of the JGA, as well as by the sympathetic nervous system and by the hormones renin and angiotensin. I'll add a couple of new hormones here, the natriuretic peptides. We can also control reabsorption. I've discussed primarily the effects of aldosterone and ADH, both of which are triggered by angiotensin II, but we'll add the natriuretic peptides here as well. Once we're done, we should know how most major diuretics function. Let me review the regulation of GFR. First off was the autoregulatory mechanism, meaning the mechanism that occurred in the kidneys themselves. The JGA can detect changes in blood pressure occurring in the afferent arteriole, which will lead to vasoconstriction or vasodilation of the afferent arteriole. A drop in blood pressure would lead to constriction of the afferent arteriole, and that would reduce GFR, helping to keep blood pressure higher, whereas increased blood pressure would lead to vasodilation of the afferent arteriole, which would increase GFR, which should lead to a drop in blood pressure. The sympathetic nervous system can also activate the smooth muscle of the afferent arteriole, shutting down filtration. Lastly was hormonal regulation. A drop in blood pressure or a drop in sodium could be detected here in the macula densa. Either of these two could lead to the release of renin, which activates angiotensin I, which activates angiotensin II, which causes body-wide vasoconstriction, and that raises blood pressure. Vasoconstriction of the afferent arteriole also reduces GFR, which should help to raise blood pressure. The effects of angiotensin II were widespread. It not only caused vasoconstriction throughout the body, it also activated the brain, triggering our thirst centers. And if we drink more, that tends to increase blood volume, which increases blood pressure. It also stimulates the release of two other hormones, aldosterone from the adrenals and ADH from the pituitary. And both of these have effects on the nephrons to increase the reabsorption. Aldosterone, of course, increases the reabsorption of sodium from the distal convoluted tubule, whereas ADH activates the aquaporins in the collecting duct, which allows water to follow that sodium. So these two hormones were very important in the regulation of reabsorption. Aldosterone activating the sodium-potassium pump, ADH activating aquaporins in the collecting duct. There are two new control mechanisms that I need to add, and those are the natriuretic peptides, called the A and B type. These come from the heart. A stands for atria, B stands for brain. Oh, well, that one was misnamed. It doesn't actually come from the brain, it comes from the ventricles, so this has been renamed B-type natriuretic peptide. But both of these have the same function. They are produced when blood pressure is too high. And when blood pressure is high, the heart has a harder time squeezing that blood into the aorta. And if the heart is working too hard, it produces these two hormones, which act to lower blood pressure at a number of different locations. The first is at the glomerulus. The natriuretic peptides lead to a constriction of the efferent arteriole and dilation of the afferent arteriole. This greatly increases the pressure inside the glomerulus, which leads to an increase in GFR, which of course means that as we filter more out of the blood, this should reduce blood volume, reducing blood pressure, reducing the amount of work that the heart has to do. The natriuretic peptides also antagonize the effects of angiotensin II, which blocks its ability to trigger vasoconstriction and also blocks the reabsorption of sodium and then water throughout the nephron. So the natriuretic peptides reduce blood pressure by two major mechanisms: increasing GFR and reducing reabsorption. In medicine, we'd often like to do one of these two things, either increase GFR or block reabsorption to try and lower a patient's blood pressure. The problem here is that there's a number of different homeostatic mechanisms involved, and that if you simply lower somebody's blood pressure, you might activate some of these negative feedback loops, which will raise blood pressure right back up again. Again, though, our two major targets are filtration, happening in the glomerulus. This, of course, was regulated by autoregulatory controls, hormonal controls, and neural controls. So this sucks. We're not going to do this. Instead, we're going to target reabsorption. Reabsorption can either be obligatory or facultative. The facultative reabsorption meant that I could adjust how much I was reabsorbing. These adjustments were regulated by hormones like renin-angiotensin, aldosterone, and ADH. So we're going to want to shy away from these, because if you were to, say, block this, your brain could always produce more of it. So in medicine, we often target the obligatory mechanisms, the ones that the body is not measuring and therefore never changes. A diuretic is any chemical that leads to an increase in urine production, and they fall into one of two major categories. The first are solutes that are not reabsorbed, and the second are chemicals that can block some sort of sodium transport protein, blocking sodium reabsorption in one area of the nephron. Either one of these will lead to an increase in urine production. Increased solutes inside of the nephron will tend to attract water, reducing the osmotic gradient. So more water will be retained in the nephron to become urine rather than reabsorbed to become blood plasma. And if we block sodium reabsorption, then less water will follow it. This also increases urine production, and both of these mechanisms could lead to a drop in blood pressure. For instance, glucose is a solute, and it can act as an osmotic diuretic. This is why patients with diabetes will have increased urine production. With elevated blood glucose, we're going to filter more glucose into the nephron. And if it exceeds the transport maximum in the proximal convoluted tubule, then glucose will wind up in the loop of Henle, where it acts as a solute, attracting water. So less water would be reabsorbed in the descending limb, and instead would stay in the nephron. The glucose transporters are only found in the proximal convoluted tubule. So if we exceed the transport maximum here, that glucose can attract water in all of the other places of the nephron where water might be leaving. That includes the descending limb of the loop of Henle, as well as the collecting duct. Other chemicals can act as osmotic diuretics. Any molecule that is small enough to be filtered but is not reabsorbed will behave as an osmotic diuretic. Mannitol is another example of this. So let's review really quickly what's happening here. Excess levels of glucose in the blood will lead to excessive levels of glucose in the filtrate, and the proximal convoluted tubule has only so many sodium glucose symporters. So when we overload the transport maximum, some of that glucose is going to be winding up in the loop of Henle. And if it does, it acts as an osmotic diuretic. For patients who are not diabetic, they should be reabsorbing all of their glucose, assuming they didn't pig out on candy bars the night before. We typically reabsorb all of the glucose that we eat. On the other hand, we typically eat way more amino acids than we reabsorb. So amino acids would be normal in urine, whereas glucose is not. That would be a sign of diabetes mellitus. Other diuretics work by blocking some of the transport proteins. There's a few weird ones that can block hormones. For instance, alcohol blocks the release of ADH, and that would block the aquaporins from functioning in the collecting duct. That would lead to a drop in blood pressure, at least temporarily, and that is what can give you a headache the next morning and be part of a hangover. Of course, if you replenish your fluids, that can reduce the severity of a hangover. Still, we would not prescribe alcohol as a diuretic for long-term control of blood pressure, and that's because it induces a number of negative feedback mechanisms. In alcoholics, for instance, we typically see elevated blood pressure. Alcohol is very good at dropping blood pressure temporarily, but do this enough and you'll activate some sort of homeostatic feedback mechanism which overshoots and actually leads to elevated blood pressure. So this would not be good medicine. There are some drugs that can block aldosterone. This is kind of weird. For the most part, we typically avoid any sort of mechanism that involves hormonal regulation. Most other diuretics block some sort of sodium blankety-blank symporter or antiporter protein found in the proximal convoluted tubule or the loop of Henle. For instance, caffeine can block sodium reabsorption, as does Lasix and diuryl. They target different proteins that move sodium and other molecules. Reducing sodium reabsorption should also reduce water reabsorption in the collecting duct, increasing urine production, and lowering blood pressure. Not every chemical that is a diuretic will lead to a drop in blood pressure, and that's because of homeostatic control mechanisms. For instance, water is a diuretic. The more water that you drink, the more that you'll need to pee, but that's not going to lead to a drop in blood pressure. Why not? Well, homeostasis. Drinking water will lead to absorption of that water in the digestive tract into the bloodstream, which will elevate blood pressure. This increased blood pressure is detected in the JGA, and this will trigger the relaxation of the afferent arteriole, which in turn leads to an increase in GFR. With increased filtration, will lead to more urine production and a drop in blood pressure, back down to our original homeostatic set point. So water is a diuretic, but it won't help to lower anybody's blood pressure down lower than it already is. What about caffeine? A lot of people worry that caffeine will dehydrate you. And that's true if you would eat the caffeine in powder form, but what about when it comes with a whole bunch of water as well? Caffeine does block sodium reabsorption in the proximal convoluted tubule, and that would lead to a drop in water reabsorption as well, which would lead to a drop in blood pressure. And if we stopped there, it would lower your blood pressure, sure, but it really doesn't. And that's because a drop in blood pressure would be detected by the JGA, which would lead to a constriction of the afferent arteriole, reducing GFR. And we would also release renin, which ultimately activates angiotensin II. And angiotensin II triggers body-wide vasoconstriction, raising blood pressure. And it also triggers the release of aldosterone and ADH, which increases water reabsorption from the collecting duct. which helps to bring blood pressure back up to where it should be. So if you were dying of thirst and came across a Starbucks, I would say go ahead and drink that coffee. What if you came across a bar? Should you drink a beer? And the answer here is no. Alcohol blocks ADH, and that stops water reabsorption from the collecting duct, which lowers blood pressure. That drop in blood pressure would be detected by the JGA, which could constrict the afferent arteriole and reduce GFR, but it doesn't stop GFR, so that could lower how quickly we're losing water. Renin and angiotensin would also trigger aldosterone and, well, can't trigger ADH because alcohol's blocked it. And who cares about aldosterone on its own? If we reabsorb sodium, but water can't follow because there's no aquaporins in the collecting duct, then that sodium's not going to do anything. And we're going to have increased urine production to the point of dehydration. So drinking that beer, you may drink a pint of liquid, but you'll probably urinate more than a pint because you can't produce ADH, even though the kidneys detect an abnormally low blood pressure. There's nothing that they can do when the last step has been blocked. What about these others here, like dandelion, hawthorn, and parsley? These are also diuretics, but whether they can be used to treat high blood pressure is questionable. I haven't seen any good evidence yet. Next up, I promised that I would discuss the use of diuretics in sports. Some drug tests, in addition to testing for performance-enhancing drugs or drugs of abuse, will also test for diuretics. The reason is that people can use diuretics to try and cheat the system. Many drugs of abuse are metabolized in the liver by enzymes called P450s that we're going to learn about in a couple of chapters. And the metabolites are then dumped into the bloodstream. These inactive molecules can be filtered by the kidneys, but they aren't reabsorbed, and so they wind up in urine. And this is how we can test for drugs. If somebody were to take a diuretic, for instance, one that leads to an increase in GFR, with increased filtration, we would be removing these metabolites from our bloodstream faster. This means you can clear your system out more quickly to try and pass a drug test. Hence, an expensive drug test might also look for metabolites of drugs that act as diuretics. Over here to the right, I show some of the metabolites that are detected in certain drug tests. Heroin doesn't actually do anything in the human body until it gets converted into morphine. And this typically only happens in the brain. And that's how heroin has its effects. But that morphine eventually winds up in the rest of the body, where the liver can metabolize it to hydroxymorphone, and then that chemical is small enough to be filtered but not reabsorbed, so it winds up in urine. And a few days later, it can be detected on many standard drug tests. Unfortunately, codeine can go through the same pathway. Some codeine molecules actually are converted to morphine before they have an effect. Other codeine molecules go through a slightly different pathway, but ultimately it winds up again as hydroxymorphone. So this drug test can't distinguish whether the hydroxymorphone was derived from codeine or whether it was derived from heroin. So if you were taking codeine as part of a cold medication, you're going to fail this drug test a few days later. Or even if that codeine came from a poppy seed bagel, which it does, a few days later, you're going to fail that drug test. So if you've had a poppy seed bagel and your work is going to test you for drugs in a couple of days, well, you might take a diuretic to try and clear these metabolites from your system more quickly so that your urine is clean when you have to go in and do the drug test. So those are the two natriuretic peptides, the A and B types. These were the only two hormones that I discussed that could lead to a drop in blood pressure. Every other mechanism that we talked about led to elevated blood pressure. Conversely, when we want to try and lower blood pressure, we use chemicals called diuretics. And these fell into one of two major categories: the osmotic diuretics, which were chemicals that were small enough to be filtered but not reabsorbed, and the loop diuretics, which would block one of the sodium co-transport proteins, blocking sodium reabsorption. That's it for all the really small stuff. Let's take a step back and look at kidney function as a whole. We might want to know how well the glomeruluses are filtering the blood. However, we don't have glomerometers or anything else to stick into a kidney to measure glomerulus function. So, to measure how well the kidneys are filtering the blood, we must measure this indirectly by doing either a creatinine or an inulin clearance test. We're going to give our patient a milkshake that contains creatinine, for instance, at a certain amount, and then have them drink it and wait a while and see how quickly that creatinine shows up in the urine. We know that creatinine is filtered but not reabsorbed. So if a patient's GFR is really low, that creatinine would not be winding up in the urine quickly. One problem with this is the body can produce some creatinine on its own, so a more accurate test would be an inulin clearance test. That way you could be sure that the inulin showing up in the urine was inulin that they drank previously. Unfortunately for this one, I'm seeing inulin used more and more as an additive in low-calorie food because the body doesn't metabolize it or use it. It does wind up in the bloodstream, but after the kidneys filter it, they don't reabsorb it because it's not a useful molecule. If a patient's kidneys were failing, you might have to put them on dialysis. Otherwise, things that the kidneys were normally removing from the bloodstream could build up. That includes toxins, for instance, bacterial endotoxins. We have a number of bacteria living inside of us, some of which produce molecules that would be damaging to us if our kidneys didn't remove them. With kidney failure, these toxins can build up to high enough levels to start doing damage to the body. The kidneys also remove excess nutrients, especially minerals and electrolytes. Without proper kidney function, some minerals could build up to toxic levels fairly quickly, like aluminum, copper, or fluoride. These are things that our kidneys are normally keeping at low enough concentrations to not be harmful to the human body. In the absence of functional kidneys, these electrolytes and molecules must be removed. And to do that, we can do either hemodialysis or peritoneal dialysis. Hemodialysis is functionally fairly straightforward. It replaces the glomerulus part of the kidneys, but not the rest of the nephron. Blood can be forced through tubes that have tiny holes. These tiny holes allow small substances to exit, but not large ones, like albumin and red blood cells. This means we're losing all of the toxins and nutrients from the blood. Therefore, we must replace all of this fluid with a saline solution that contains enough nutrients and electrolytes. This is mixed back in with the cleansed blood and returned to the body. This water that the Ringer's solution is made from must be ultra purified. We can't just use regular tap water because any amount of aluminum or fluoride in it could easily reach toxic levels if placed back into our patient's body. To actually do hemodialysis is a little bit trickier than I just discussed. First off, if we're going to be removing blood from the body by inserting a needle into a vein 3 times a week, that vein is going to collapse pretty quickly. So we must strengthen up a vein for usage first by creating a fistula. An artery and a vein are sewn together, and after a little bit of time, because that vein is now under higher pressure, it's going to do what any muscle-containing organ is going to do. It's going to get stronger. The smooth muscle becomes thicker, and eventually now we can stick a needle into that vein to remove the blood from the body, purify it with the hemodialysis machine, and then return it. We must also use agents that block blood clotting, so heparin must be given to our patient, otherwise the dialysis machine would clog. And as I mentioned earlier, this water must be ultra purified. We have to make sure that the only things we're returning to our patient are the red blood cells and large proteins that didn't get filtered by the dialysis machine. Plus, we're returning a bunch of nutrients that we added to our ultra-purified water. If a patient can't go into a hemodialysis clinic 3 times a week, for instance, maybe they have a job that they need to keep their health insurance, to pay for dialysis, we're going to opt for peritoneal dialysis, which technically is less efficient than hemodialysis, but seems to be just as effective. Rather than cleaning toxins out of the blood, this will clean toxins out of peritoneal fluid. Any wastes or toxins that wind up in blood, some of those will wind up in the peritoneum. So if we remove this fluid and replace it with new fluid, that will help to remove toxins from the body. And the reason this is easier for many patients is that it can be done at home once this little tube has been inserted into the peritoneal cavity. It's also significantly cheaper than hemodialysis. So if you do any sort of medical outreach with disadvantaged populations or traveling to other countries and you need to do dialysis, you're probably going to be doing peritoneal dialysis. Let's return to a functioning kidney. That urine is produced in the nephrons, most of which are out in the cortex, but some of the juxtamedullary nephrons do dive deep down into the medullary region, back up again, and then the collecting ducts go down deep once again. All of these collecting ducts will empty their filtrate into a minor calyx, and at this point we would start calling it urine. We're not going to reabsorb anything more from the calyxes. And this urine would then travel down to the bladder for temporary storage until we're ready to excrete it by the process of diuresis. Urine is a clear solution, and it should be sterile. It's yellow because of a molecule called urobilin. This is a breakdown product of our red blood cells as they're being recycled. You will learn a lot more about the normal and abnormal components of urine in the lab portion. I'm going to skip ahead to the rest of the urinary tract. From the kidneys, urine will travel down these ureters to be stored down here in this bladder, and ultimately urine could exit via the urethra. Starting from the minor calyxes, we're going to be lined now with transitional epithelium, all the way down to the bladder. This transitional epithelium is specialized in that it can stretch and then contract back to its original shape. We're going to need to stretch this tissue when it's full of urine, but when we're not producing urine, it needs to contract. The ureters are also lined with smooth muscle, and they travel all the way down to the base or the bottom of the bladder. This seems like a strange place for urine to enter the bladder from the bottom. It'd be like trying to fill a cup from the bottom of the cup rather than pouring water in through the top. To prevent urine from flowing backwards, the entrances are slit-like rather than round, which to me seems like a bit of a kludge. It would have been a lot better just to attach these ureters to the top, but they're not, and they're down here at the bottom. And the reason that they're at the bottom, I can't explain to you until we reach the chapter on developmental biology. So for now, this is just kind of weird. The ureters are lined with transitional epithelium from here to here. They then have a bunch of muscle tissue as well. And anchoring all of this together are bits of connective tissue. We see some submucosa here, but there would also be an outer layer of connective tissue out here. The smooth muscle generates what are called peristaltic contractions, which helps to push urine towards the bladder. If we're standing upright, gravity can do most of the work, but when we're laying down, it helps to have these muscular contractions forcing urine towards the bladder. The bladder has just one function, which is to temporarily store urine. It can store about a liter of urine at a time. Seems like that's been the answer for everything in this chapter, 1 liter. It's anchored into the pelvic cavity by a number of ligaments. The mucosa, which is transitional epithelium, has large visible folds called rugae. These folds will stretch and disappear when the bladder is full, but then the inside of the bladder appears wrinkled when it is empty. Except for at the base of the bladder, this region called the trigone that is delineated by the 2 openings for the ureters and the 1 exit for the urethra. This region is smooth and shaped like a funnel down towards the urethra. One of the first chemicals that was identified as a carcinogen are these nitrogenous yellow dyes shown over here to the right. This was a big deal in medicine because previously it was thought that people got cancer simply because they were bad people. I'm not really making that up, if I'm kind of simplifying it a bit. So this led to a major change in how we thought about cancer. That maybe you could avoid at least certain types of cancer if you avoided certain things in the environment. These azo dyes were used in the garment industry to dye clothing yellow. And the people who did the dyeing of the clothing often did the dyeing of bladder cancer. That's because these chemicals are small enough to be filtered, but they're not reabsorbed by the nephrons. And so when these get absorbed into the bloodstream through the skin, the kidneys filter them out, which helps to protect the body, but then they wind up in the bladder for a while. So if they're going to be causing cancer anywhere, it's usually in the bladder. In the neck of the bladder is a ring of smooth muscle called the internal urethral sphincter. It's actually a continuation of the muscular portion of the bladder itself called the detrusor muscle. Here is my cartoon of a bladder in cross-section, and the internal urethral sphincter, again, would be down here. During the filling phase, this sphincter is closed. The sympathetic nervous system will cause that sphincter muscle to constrict, but also cause relaxation of the detrusor muscle. Hence, the bladder is capable of filling up with urine. We can do 2 different types of muscle actions here, both caused by epinephrine, because we've got different types of adrenergic receptors. The bladder will continue to fill up with urine until it contains about 1 liter, and at that point, stretch receptors found in the bladder wall would be activated, and these send signals to the spinal cord. This will trigger a reflex. Parasympathetic fibers will be activated in the spinal cord, releasing acetylcholine onto the bladder. This would cause contraction of the detrusor muscle and relaxation of the internal urethral sphincter. Luckily, this is not going to lead to urination yet. Otherwise, you would urinate every time your bladder reached the 1 liter mark. Instead, we're going to wait until we're ready. We can do that because we also have an external urethral sphincter, here lower down on the urethra. This muscle is under the control of the somatic nervous system, meaning we have conscious control over it. So what the reflex does is cause the bladder muscle to start contracting, the opening of the internal sphincter, and now this is going to put some force on the external sphincter. And we can feel this. This is the need to pee. We don't feel it when the bladder is 90% full or 80% full. And that's because we haven't activated the stretch receptors. So the bladder is still relaxed. It's not under pressure. It's only when the reflex is triggered and the detrusor muscle begins to contract that pressure is being created that can activate baroreceptors that signal the brain that, hey, we need to pee. The external sphincter is skeletal muscle and is under voluntary control. Luckily, it has resting muscle tone, so we don't have to think about not peeing, even though the urinary reflex has been activated. To summarize, the micturition reflex, or the urination reflex, occurs because stretch receptors are activated on the bladder, and these send signals to the spinal cord, which activates the parasympathetic branch, which causes contraction of the detrusor muscle and the opening of the internal urethral sphincter. But it's a different nerve here that controls the external urethral sphincter, and this is under the control of the somatic nervous system. In infants, this reflex isn't controlled yet, and so this can lead to incontinence. Damage to any of the sphincters could also lead to incontinence. Lastly, we also talked about the vasovagal reflex. Extreme fright can sometimes trigger a parasympathetic response rather than a sympathetic one. And with massive parasympathetic stimulation, this could lead to a massive drop in blood pressure that causes the person to pass out, and they might also urinate. Conversely, sympathetic stimulation would not cause you to pee your pants, because sympathetic stimulation causes the bladder to relax and the internal sphincter to close off or constrict. That's why if your patients are nervous at the hospital, they may have trouble giving you a urine sample. Their bladder is staying relaxed because of their sympathetic tone. The bladder is lined by transitional epithelium. We then have a submucosa of some connective tissue, and then we have the detrusor muscle, which is a few layers of smooth muscle here. Historically, if a patient needed to have their bladder replaced, maybe because they had bladder cancer, or maybe because of trauma, surgeons would cut out a small portion of the intestines and sew it into a new sac that could form a new bladder. The problem with that is that the intestines are pretty good at absorbing things, and so some of the waste products that should have stayed in the bladder tend to get reabsorbed by a fake bladder made from intestines. A better and newer procedure is to grow a new bladder. You can get a bladder from a cadaver and strip it of all of its cells, leaving behind just the scaffolding or the connective tissue. And then, gathering stem cells from your patient, you can drop those stem cells onto this bladder scaffold, and they will grow into new smooth muscle and transitional epithelium. Because the cells of this new bladder are the patient's cells, the risk of tissue rejection is minimal. From the bladder, urine will be excreted through the urethra, which exits the body. The urethra is also lined with transitional epithelium, shown here in this darker purple, and then a little bit of connective tissue, then we have muscle, and then an outer layer of connective tissue as well. So that wraps up the urethra and the bladder and the ureters, our tubular organs of the urinary tract. And this represents the end of the lecture portion of the urinary system.